So in lab this week, you carried out a reaction and made some products. This video is going to go over how to analyze the products and how you can use the mixtures of the products to figure out what kind of mechanism that the reaction went through. So the type of reaction you carried out is what's called a nucleophilic substitution. We abbreviate this with a capital S for substitution. We put an N down below to represent the nucleophile. And then there's going to be a number that goes after it, which we will figure out in the course of this video. So essentially what this reaction involves is an alcohol, and the alcohol reacts with a halide ion. The halide ion has electrons that it donates, and so a substance that donates electrons is known as a nucleophile or a nucleus lover. And then what we end up forming are an alkyl halide. So basically the halide takes the place of the OH group on the alcohol and water. So in lab, you actually worked with two alcohols. The first one was known as N-butyl alcohol. It consists of 4 carbons attached to the OH group. The OH group is sometimes called the hydroxyl group. And the name of this is N. N just means that it's quote-unquote normal, which means it's in a straight line. Butyl refers to the fact that it has 4 carbons, and alcohol means it has this general structure. The other alcohol that you worked with, it has the same number of carbons, but they were arranged a little bit differently. The name of this one is T, which stands for tertiary butyl alcohol. Okay, and then in terms of the halide ions, you also worked with 2 of them. We had bromide, Br-, and chloride, Cl-. And so basically we're trying to figure out what kind of mechanism this goes through when the halide attaches itself to the red part of the alcohol. So the next step is to talk about the possible mechanisms that this reaction can go through. So, as you know, mechanisms are the steps of a reaction, because most reactions don't happen in one step. No matter what, the reaction has the first step that's relatively fast, and what happens to it is basically we start with the alcohol. So this is our alcohol, and we add a proton to it. So if you remember, one of the first things you did when you carried out the reaction is you measured out volume of concentrated sulfuric acid. So that was a rich source of protons. And what happens is the H plus gets attached to the oxygen. So those electrons get shared, and what we end up with is just simply the alcohol with an extra proton on it. Okay, and this has the name of a protonated alcohol. So no matter what the mechanism ends up being, this is the same first initial step, and this step is fast. So it's not the rate determining step. In terms of the rate determining step, RDS, there are 2 possibilities. And essentially the difference between the 2 is whether or not the halide is involved in the rate determining step or not. So for the first possibility, it's going to be a unimolecular rate determining step, which means that it's going to involve just one particle, one molecule. So in this case, that would be the protonated alcohol. There it is. And then essentially what happens here is the electrons in this bond right here transfer over entirely to the oxygen. So what we're left with is the R group, which now has a positive charge. And then we have, if you look at the rest of the molecule, we have oxygen with 2 hydrogens, we have ourselves water. So the R plus particle has a name. Remember, this group is made of carbons and hydrogens, and so this group is what's known as a carbocation. A cation just means it has a positive charge. So that's unimolecular. The other possibility is if the halide is involved, and this would be a bimolecular process. So we have the protonated alcohol again. And now we're going to involve the halide ion. And so what happens here is essentially the same thing that happened in the other step, but at the same time, the halide attaches itself to the R group. So basically what happens is the electrons in this bond go to the oxygen, and then because the halide has an excess of electrons, it's going to attach itself to the R group by donating its electrons, which is what we call a nucleophile. So what we end up with is the alkyl halide and water. Okay, so now we have 2 possibilities here. Remember that the type of the reaction we were looking at is a substitution, and it's nucleophilic. When it is unimolecular, that means the rate determining step happens in one step, so involving one molecule. So this is called an SN1 reaction. When we have it being bimolecular, where 2 particles involved in the rate determining step, this is called an SN2 reaction. So now, the way we're going to tell how this happens is the mix of products that we're going to get. So if the halide is involved in the rate determining step, so remember when you carried out the experiment, you had in the containers, you had both halides present. You had both bromide and chloride present. And so one of these is going to be able to do this process faster than the other. So what this is telling us is that there's going to be a preference for one of these products or the other. So in this case, what we would end up with is not a 50-50 mix of products. Meaning not a 50-50 mix of RBr and RCl. So if we look at our products and we don't have a 50-50 mixture, then that means that this is an SN2 reaction. On the other hand, if in the unimolecular step, if the halide's not involved, it's not going to matter which one has preference, because neither one are going to matter because they're not part of the slow step. So in this case, if X minus is not involved, then this is going to be a approximately 50-50 mixture of the alkyl bromide and the alkyl chloride. So in this case, if we get about a 50-50 mixture, then we can say that it's probably SN1. If we don't have a 50-50 mixture, then it's likely that this is an SN2 reaction. Okay. So let's talk about how we analyze your product and the instrumentation that we used to do that. So in the lab, you carried out 2 reactions. One of them involved N-butyl alcohol and the other involved the T-butyl alcohol. In both cases, you had an equimolar mixture of the two halides. So you had Br minus, Cl minus. Equimolar just means that there is the same number of moles, basically the same number of particles of bromide and chloride. That's what's important. And then the same story on this one. So the N-butyl alcohol is the one that you carried out in that big complicated reflux apparatus. So this was in this round bottom flask that you heated and refluxed for about an hour. The other one didn't need that level of involvement in the reaction, so you basically just carried out this one in a centrifuge tube. And then you ended up with isolated your products. You put them in a little test tube. So here's your products. And then this one gets sent to a gas chromatograph, or GC. And then for this one, the products got sent to nuclear magnetic resonance, or NMR. So now these technologies, GC and NMR, are both pretty complicated, but I'm just going to give you a surface level understanding of how they work. So quickly, we're going to talk about GC and NMR. GC again stands for gas chromatography. And NMR stands for nuclear magnetic resonance. If you have to take organic chemistry, you'll talk about these technologies in detail. But essentially, what they both are going to be used for in this case is to separate and quantify the products. All right, just a quick overview of how gas chromatography works. If you've seen in the back room of the lab, there is an instrument. There are actually two big instruments back there. One of them is a gas chromatograph. And basically the way it works is we use a gas to push our sample through a column, and then the column separates the two products, which then get detected and turned into an electrical signal. On a smaller scale, we have our liquid sample right here, which has a mixture of different substances. You inject it literally with a syringe into the instrument. And what happens is the instrument heats it up to vaporize all the components. All the while, there's a gas, sometimes helium. It depends on what you're working with, but this one gets pushed along with helium. And as it's getting pushed along, these are getting separated from each other. So you can see that now we have them going through the column at different rates. The blue one's going through the fastest and the yellow one's going through the slowest. And then at the end, they get detected and turned into electrical signal. And so what that's going to tell us is how many components are in the mixture and their relative amounts. So that's the quick version of how gas chromatography works. So what we end up with, the readout that we get, is a graph. On the y-axis of the graph, we have this unit, PA. So you'll see this on your own GC readout. What this stands for is picoamperes. It's just a unit of electrical current. And so you're going to get these peaks, and the higher that they are, the more of current they made. And so if there's more substances, that's going to make a bigger peak. On the x-axis, we have time. So this is literally how long it takes to come out of the column. And in this case, it's measured in minutes. And so what we end up with is we get a line that has some peaks on it. So if our mixture contained two substances, that would correspond to two peaks here. And then what we can do is we can have the computer measure the area under the curve. And what the area will tell us is the relative amounts of the two components of here. So if the area ends up being, of this one ends up being 2 times bigger than the other one, it means that we made 2 times as much product here than we did for this one. So that is a crash course in gas chromatography. Let's quickly, even more quickly, talk about nuclear magnetic resonance, NMR. So the short of it is the fact that the nuclear magnetic resonance has the word nuclear in it. And what that's doing is it's referring to the nucleus of the atom, positively charged core. And what nuclei do is they have this characteristic to them that we call spin. And so when you have a spinning charged particle, what you end up with doing is you make a magnetic field. So you can think of the nucleus here like a small magnet. So it'll be south, and then we'll have a north pole. And so what we do with our samples to analyze them is we put them, here's our sample in a test tube. We put it in the presence of a very strong magnetic field. So this is a magnet here. The stronger the magnet that you use, the better you can read into the signals that you get. And so essentially what happens is we bombard the sample with radio signals. And depending on the frequency of radio signals, the sample will emit radio signals of a particular frequency. And then that will be plotted on a graph as a signal, very similar to what the gas chromatograph graph looks like. So we end up with another graph. The units on the axes are not terribly important, but the important part is that we end up with a graph that has signals on it that corresponds to the products. And just like before, we can make conclusions about the relative amounts of the products based on the area underneath the peaks here. So essentially, these two instruments are going to tell us what the relative amounts of the products are. So let's now look at an example of the GC. When you get your GC results, the first page is going to be a specific standard that was injected into the GC. So if you look at the top here, it says 3:1. Basically what this is we took a mixture of the products that was known to be a 3:1, just so that we could see what kind of retention times we get. So we have our peaks here. You can see that this one corresponds to N-butyl bromide. This one corresponds to N-butyl chloride. And then down at the bottom, there is a table where it talks about the relative areas here. So if we inject one that has a ratio of 1:3, then these are the relative areas we would expect. We'd expect a 1:3 ratio. On the second page of yours is the mixture, your actual mixture that you made in lab. And so here's a sample one. So again, you can see that the N-butyl bromide had the retention time right here, 0.293. The N-butyl chloride had a retention time of 0.213. And if you go back to the first one, you can see that they overlap decently. So the chloride is 0.213 in that one, and it's 0.213 in the in yours. So that helps you to say that, well, if they have similar retention times, it's probably the same substance. So in this one, what we can see is if we look at the relative areas, we can see, and if we take a step back, recall that what we're looking for is the mixture ratio, is it 50-50 or not 50-50? So the exact ratio that you get here isn't as important as the fact that it's in this case it's not 50-50. So that's what's going on there. So that implies something about the mechanism. So you may have to go back earlier in the video to see whether that corresponds to SN1 or SN2. Okay, and then the third page of your product is going to be the NMR, and this is going to be for the T-butyl. And so what you'll end up with are two peaks because we made two products here. And you'll see that these numbers here are a calculation of the area under the curve. And so what you can get from this is that these two are approximately equal to each other. So in this case, they're approximately equal to 50-50. And again, from that, we can determine whether or not the mechanism is, the reaction is SN1 or SN2. Okay, so this concludes the video. Hopefully this will be enough information for you to complete the post lab. Have a great